He will be presenting the topic of spatial profiling of the tumor microenvironment to identify biomarkers of therapy. He is from Queensland University of University of Technology of Australia. So good late night to you, and I pass the virtual mic to you.
Perfect, thank you, Pani. And can you hear me and see my slide okay?
Yes.
Yeah, perfect.
Yes.
All right, I'll kick off then. So hello everyone. It's almost midnight in Brisbane, and I'm grateful for this opportunity, and then thank the committee for the invite. I sort of jumped in and out of a couple other talks a few days ago, and it seems to be a really engaging meeting. So I'm really excited to be a part of it. So I'm a research fellow based at the Queensland University of Technology, and my work is looking at spatial biology approaches for identifying better sort of predictive models for checkpoint inhibitor therapy. So it's a sort of, it's leaning towards immunotherapy. So this was a photo taken last year at AACR in Atlanta, where Jim Allison gave his talk about CTLA-4 work. And it sort of just reminded me what physical conferences looked like, you know, a year ago. And so Jim Allison's work sort of led the field with CTLA-4 treatment for immunotherapy. And one of the really unmet clinical needs in this space has been identifying which patients would respond to therapy. And they seem to be really effective in a subset of patients. So in non-small cell lung cancer, head and neck, as you can see in this chart up here, triple negative breast, it's this field sort of being led by melanoma. So in a subset of patients, they tend to do really well. So identifying those patients is critical. And then in some of the patients, they have a durable response and long-term benefits from immunotherapy compared to standard chemo. So one way to do this is to look at a tumor mutation burden. Or essentially, and now there are drugs such as pembrolizumab, which are given based on, and it's pan-tumor, so it's you know, it's tissue agnostic, and it can be given when your mutational load is above 10 mutations per megabase. It used to be 20, but it's been brought down to about 10. It is quite arbitrary at the moment, so I think they are trying to refine it. So TMB is one way you can tackle this. But the other way, which I think is more nuanced, is looking at the tumor microenvironment and trying to characterize this at a greater depth. So what's really challenging with the tumor microenvironment is trying to get a handle of the cells and the immune contexture within it, to understand the interactions, the activation status, the cell types that are in the tumor microenvironment. But I think with advancements in technology, now we have the tools to interrogate the tumor microenvironment at a greater level than been previously possible. So one way we've been able to do this is using this Nanostring digital spatial profiler. And what this essentially does is it uses antibodies, and the antibodies are, they're coded onto your tissue, and they're tagged with UV photocleavable linkers essentially. And when UV light is shone above these regions of interest, these oligos are liberated. So your tissue is left untouched, and you're able to interrogate different areas within the tissue with spatial resolution. So you could typically do about 80 to 100 proteins on a single slide. So imagine trying to do that on, you know, serial slides. It would take up an entire block. So you could, the beauty of the technology is you can do it on archival blocks, 20 years old, and just freshly sectioned. And you could look at about 80 to 100 proteins spatially, and between 96 to about 20,000 RNA targets, a whole transcriptome. We have done some 1800 plex assays, and we are beginning whole transcriptome assays in the next couple of weeks. So again, it's your RNA tagged to a UV photocleavable linker, which is liberated when light is shone above it. So this is essentially the workflow. You stain your single slide. So it's a single, you know, unstained section. You define your regions of interest with a pathologist. With, you know, for cancer, this is pretty straightforward, and I'll talk about some of the work we've been doing for COVID later on, which might, you know, it might be timely, so I just put those slides in. And then you're able to define regions of interest and liberate the signal from those regions of interest down to about 10 cells using these mirrors and UV light. Those are counted. So the oligos are liberated, they're counted, and you get digital expression data per region of interest. So this was a review paper we wrote with a student that's hopefully going to come to my lab and from Iran, and Prof. Razai. And so this is what it looks like. So you've got your H&E image on the top. In the middle, that's your multiplex immunofluorescence for morphology. So you've got your pan-cytokeratin, CD3, CD8, DAPI. So that's just showing you what your morphology looks like. And within the tissue, then you can go choose 12, 24, 36 regions of interest, and then get that. So this assay was looking at proteins, and we were looking at about 60 proteins within those regions of interest. We tried to get exome data on all these patients by sequencing the tumors, but we didn't have sufficient tissue available. So I think this is one way there was one patient where we were able to get a TMB measured, but obviously need quite a bit of material for that measurement. And what you're able to do is you're able to go through the tumor and essentially get a snapshot of the signal that's coming from that region of interest. So you can look at your tumor infiltrating your TIL areas, your tumor bulk, your stroma, your normal tissue, and get expression data for those regions of interest. And you can, I'll show a bit later, you're able to mask on different cell types. And what we can see for head and neck cancer here is some other targets, not just your PD-1, PD-L1, but other immunomodulatory targets that are being looked at. So VISTA, STING, IDO1, IDH, these can be measured quite quickly and efficiently just from a single piece of tissue. And this is what it essentially looks like, the workflow. So you'd go through your tissue, you'd stain it with these morphology markers, but then these are at the same time, they're stained with about 100 other proteins, or you could do RNA targets as well. And then you'd go define different regions within that tissue that you'd want to liberate the signal from and count that signal digitally. So this was an example of a head and neck patient where they were given immunotherapy in the metastatic setting, so NIVO in the metastatic setting, so after multiple rounds of chemoradiation. For head and neck cancers, it's given in the metastatic setting or now in the recurrent setting. And this is what you're able to do. So look at 12 regions of interest within this tissue, and these are the markers that we're profiling. So you're profiling across immune activation, immune cell profiling, drug targets. So you could look at all these other immunomodulatory targets, STING, IDO1, VISTA, OX40, and get that signal per region of interest and then measure across the tumor and see what that looks like. And we're using this technology as a discovery tool at the moment, and then trying to validate it using whole slide multiplex IF, about 9 colors at the moment. We have done some orthogonal validation using Opal Vectra, where we've done similar regions of interest between the two systems, Nanostring and the Opal, to make sure that we are getting similar readouts on both platforms. Obviously, it's a bit challenging to get the exact same regions of interest because they are in serial sections, but we're pretty close to that. And what you're able to do is then compare your tumor signal versus your immune or your stromal signal, as well as your TIL, the infiltrating edge versus the immune regions within that tissue. And from this initial study, what we found was, the hypothesis we went in was, or the theory that was around at the moment at that time, was that CD8 T cells within the tumor would be predictive of outcome to immune checkpoint inhibitor therapy. But what we found from this initial cohort, where each dot represents a region of interest, was that CD8 was not associated with progressive disease or non-progressive disease. But we did find that there were certain other markers such as CD44, STING, CD66B, phospho-AKT, and P10, which did associate with a progressive phenotype. So what we're now trying to do is to look at this spatially and now get a readout for the whole slide assessment of these markers that we could see were differentially expressed. And this is just in lung cancer. This is an expanded panel. So you're able to do tissue microarrays as well. So this is about, I think it's about 100 cores on this TMA. and you're able to go in and mask. So what you can do on this is your panel of proteins are on the left here, but in the middle, what you can see is you're essentially calling the cytokeratin positive area as based on morphology, and you're able to get the cytokeratin area in its totality liberated and the signal from that and create a tumor mask. And when you do the inverse, you get the totality of the remaining region or the microenvironmental mask. So you get 2 signatures per core, and this is a strategy that we try to use on this TMA. And we've got a preprint on this at the moment. And what was interesting was that we could see now, you know, the markers that are associated with progression, and you could see which sort of, you know, the markers in the immune compartments, what the markers were that associated with the worst outcome, and those that were in the tumor that associated with a better outcome. And I'll just quickly go into some of the COVID work because it's timely, and I think that, you know, it would be interesting to share. What we had were rapid autopsy samples from COVID patients collected from Brazil. Lung, myocardial, we've got placental material as well, and trying to look at vertical transmission. And this was an interesting study because there was an Ackerman et al. Paper in May in the New England, which did bulk gene expression analysis of autopsy samples comparing COVID versus pandemic H1N1. What we did for this cohort was we had COVID, pandemic H1N1, the 2009 strain, and the normal healthy lungs. We ran the H&E, Mason's trichrome for fibrosis, IHC for the spike, RNAscope for the virus, path review, and then the Nanostring 1900 plex spatial assay plus spatial bioinformatics on top of that. So again, it was 3 TMAs, COVID, H1N1, and normal, where each core represents 1 patient. And what's really interesting with this is that you're able to define different tissue types. So it's not a mashup of your tissue. You can actually demarcate different different tissue types based on your pathology review of that. So this is just a snapshot of what we had in the lab. So we had, you know, the multiplex IF, the IH, the Mason's trichrome, the H&E, and we had 1 more monitor for the RNAscope, so we could then overlay the cell types. So we could identify the hyaline membranes, the type 2 pneumocytes, the bronchioepithelial cells, and what you're able to do is go through each core and liberate the signal. So you can custom draw around your regions of interest. So if you had a bronchiole such as this region in here that's shown in a square, what you're able to do is to draw around that bronchiole and then liberate the signal of that and get about 2000 plex RNA transcript data on that. And what you're able to do is overlay the RNAscope images over this to see where the virus is binding to. So these are just PCA plots by disease type. So this is a helicopter overview of your data set, your transcript level data. So what you can see here is, so this is looking at your 2000 plex RNA signal, you know, without any bias. And you're just pulling the data in different ways. And what you can see at the top is you've got your COVID in the blue, in the red is your pandemic H1N1, and then the normals in the yellow. So you see good separation of your COVID from your normal, which is reassuring. And then what we do is we identified it by the cause or the patient sample. So now you've got the spread of the patient samples across it over these. And you get sort of 2 different populations. There's this one population on the right, and there's this one on the left. So at the transcript level, we can't really tell what this is, right? But when you overlay the pathology over the transcript data, now you can see the cell types separate out your data set. So you can see the bronchioepithelial cells are at the bottom left here. Your type 2 pneumocytes are in the light blue. The macrophages are in the brown, and your lymphocytes are in the green. So now you can see the beauty of the technology because you're marrying the pathology data with the transcript level data. And then we ran the differential analysis of it. So COVID versus control, 2000 genes, spatial again, and COVID versus H1N1. And what's really interesting is this, one of these key genes, and the virologist just really got really excited when she saw this, was that IFI27 was elevated in COVID versus control, and IFI27 was again elevated in COVID versus H1N1. So IFI-27 is known to be elevated in peripheral blood of COVID patients and in the nasopharyngeal swabs of patients, but there haven't been studies demonstrating where this was coming from. So we believe this is one of the first studies demonstrating that IFI-27 is actually coming from the lungs, and we've actually done some follow-up work on this, and we can see that it could potentially be a triage biomarker for COVID-19. We see that it's highly elevated in severe cases, and then it sort of drops down and you don't pick it up in the healthy cohort. And we've done some high virus versus low virus as well, and we've got myocardial data which has come through recently. So with that, I'd like to thank the team that's been involved in this work, and I just direct you towards this preprint that we've put out on this COVID work. But again, yeah, please reach out, and thank you for the invite.